because of the jointed legs. It belongs to the subphylum Mandibulata because of the mandibles, presence of mandibles. It belongs to the class Crustacea because of the respiratory organs as gills. Then it belongs to the subclass Malacostraca because it is having 19 pairs of appendages, of which the 13 pairs belong to the cephalothoracic appendages. Then again, presence of compound eyes with stalk. Then it belongs to the order Decapoda because of the presence of 5 pairs of walking legs, and it belongs to family Palimonidae, genus Palimon, and species Malcolmsoni. It is also called the Macrobrachium malcolmsoni sometimes. Now, prawn respiratory system can be studied under 3 steps. One is respiratory organs and blood supply to respiratory organs and mechanism of respiration. In these 3 steps, we are going to discuss prawn respiratory system. First of all, respiratory organs. Actually, prawn has 3 types of respiratory organs. Mainly, the main respiratory organs are the gills, whereas the branchiostegite and as well as epipodites are also functions as, to some extent, respiratory organs. And before going into the blood supply to respiratory organs, let us discuss the respiratory organs in detail. Now, coming to the first organ of the prawn respiratory system, just now we have seen this branchiostegite, and this branchiostegite is each gill chamber in the prawn is enclosed between the branchiostegite, or which is also called gill cover externally and the wall of the thorax internally. Here you can see this is the outer covering. This is called the branchiostegite. Actually, this branchiostegite is formed by the hanging down or lateral hanging down of the carapace, and in the last lessons we have seen how the body of the prawn, structure of the prawn. The body can be divided into the head, thorax and abdomen, and head and thorax are covered by a shield like structure that is called carapace, and this carapace laterally hangs down, and this hanging down portion of the carapace is called as branchiostegite. And this branchiostegite, inside of which it covers the thoracic region, and in between the branchiostegite and thoracic region, this portion is called as gill chamber because here the gills are present. Gills are attached to the thoracic wall, so they are covered by the branchiostegite, which is an extension, lateral extension of the carapace. And this branchiostegite encloses the gill chamber, and whereas this branchiostegite, whose inner lining is highly vascular, the branchiostegite's inner lining, so this inner lining is on both sides is highly vascular, that means supplied with blood vessels, blood capillaries, and forming, that's why form a good respiratory surface. So actually the prawn has main respiratory organ, just now we have discussed, is gills, but in addition to that, branchiostegite with because of the having the supplied with the blood vessels and blood capillaries also provides respiratory surface. So an amount of, or a considerable amount of gaseous exchange also takes place in the wall of the branchiostegite, inner wall of the branchiostegite. Then so when water flows are touching the branchiostegite, inner wall of the branchiostegite, here the dissolved oxygen from the water is absorbed into the blood that is present in the blood capillaries that are present in the inner wall of the branchiostegite, and in exchange they give out carbon dioxide into the outflowing water. So this is about the branchiostegite, so which covers, here you can say branchiostegite is an extension of lateral extension or lateral hanging down portion of this carapace here. So So this is the, you can see carapace. So it is hanging down laterally, forms the Branchiostegite. Now coming to the next portion, the epipodites. Epipodites. Second one, then Branchiostegite, after that epipodites. Where are these epipodites are present and how many of them? Epipodites are actually 3 pairs of epipodites are present. And these are just outgrowths. They are simple leaf-like outgrowths of the integument, outgrowths of the integument. And whereas what is the main feature of the respiratory organ? It should be supplied with the more number of blood vessels. So that's this is called highly vascular. So as these epipodites are also supplied with the high number of blood vessels or blood capillaries, they also help in gaseous exchange to some extent. So where these epipodites are present, they arise one pair each from coxal segments of each pair of maxillipedes. Now you can see here in this figure we can have a look at the maxillipedes. So this is the first maxillipede, and where epipodites are present here, a pair of epipodites are present here, and second maxillipede, this is the second maxillipede, and these epipodite is present here. Another side, one more, and then third maxillipede, this is the third maxillipede, and here this is the epipod. So just now we have seen the epipodites. These epipodites are 3 pairs of simple leaf-like highly vascular outgrowths of the integument. They arise from the coxal segments of the 3 pairs of maxillipedes, and they are located in the gill chamber just beneath the scapognethite of the maxilla, and they function as respiratory organs like that of the primitive gills. Let us discuss now the prawn actual respiratory organs of the prawn, which are called as gills. And there are 8 pairs of gills. There are 8 pairs of gills in the case of prawn, which are located in the gill chamber. And just now we have seen the gill chamber. Gill chamber is present in between the thorax and as well as branchiostegite. Branchiostegite is nothing but lateral hanging down portion of the carapace. So between this the branchiostegite and thorax, there is a gill chamber is there. In the gill chamber, these gills are located, and they are 8 pairs in number. 8 pairs. But when we open the branchiostegite and see, we can see only 7 pairs are visible. Only 7 pairs are visible. And one gill pair, where is this one gill pair goes? And this one gill pair is present hidden behind or beneath the second gill. Now we can count the gills here. So the first pair of gill, second pair of gill, third pair of gill, fourth pair of gill, fifth pair of gill, sixth pair, and seventh pair. So what happens to the 8th pair? 8th pair is present behind the second pair. So this is the second pair, and we can have a look at the, we can see you can see here. Now we can have a look here. A different color is there, so we can differentiate this. Behind this second pair of gill, there is one more gill is present, and this is the 8th pair. Maybe it is a third pair. So like for example, this is the first one, this is the first one, second one, third one, fourth one, 5, 6, 7 and 8. Okay, so all these are in line, they are arranged. All 8 pairs of gills are present. On opening of the branchiostegite, only 7 pairs can be seen visible clearly, and the other pair, on one pair, is present behind the second pair of gill. Now then there are all these gills, though they look like same, but based on their position in relation to the thoracic appendages, they are categorized into 3 types. So what are they? Let us see. So they are categorized into 3 types based on the place of origin and attachment to the body. First one is the podobranch, which is called food gill, the first one. So it is in a serial order something fishy happened. Now here let us have from this is the first one and this is the second one and this is the third one in serial order. So first one is podobranch, which is called as the foot gill, and it is the gill attached to the basal podomere which is called as coxa, So this is here you can see the podobranch. So podobranch or foot gill is the first type of the gill. It is the gill attached to the first podomere or coxa of an appendage. That appendage is second maxillipede. First coxa of second maxillipede, this podobranch is attached. This coxa here. This is the second maxillipede, and this just above the second maxillipede, this podobranch is attached. Now coming to the second one, arthrobranchs. This arthrobranch, they are two pairs in number. As we have seen, these are the arthrobranchs, and that means second one and third one. So second and third pairs of the gills, which are called as arthrobranchs, are otherwise called as joint gills, and these are the gills attached to the arthrodial membrane that connects the thorax with that of the appendages, and that appendage is called as third pair of maxillipede. You can see this is the third pair of maxillipede, and attached to that an arthrodial membrane is present. That arthrodial membrane attaches these gills to the thoracic wall. Then coming to the other type, third type, and these third type are called as pleurobranchs, and these pleurobranchs are otherwise called as side gills, and they are 5 pairs in number, and these are attached to the lateral walls of the segments to which the appendages belong, or you can say they are attached to the inner wall of the thoracic parts, and those thoracic segments to which these 5 pairs of walking legs are attached. So this is the first walking leg, second walking leg, third walking leg, fourth walking leg, and fifth walking leg, and these walking legs which are attached to those segments, and inside the wall of those segments these gills are attached. So these are called as pleurobranchs or side gills. There are 5 pairs. Now, so just now we have discussed prawn respiratory system. The gill types we have discussed. The podobranch, which is called foot gill, is one pair which is attached to the coxa of each second maxillipede. Attaches. Second one is joint gills or arthrobranchs. They are 2 pairs in number. They are attached to the arthrodial membrane connecting the thorax with the third maxillipede. Just now we have seen. And the pleurobranchs. So here also the same figure we can see here. The pleurobranchs, which are also called side gills, they are 5 pairs in number, and these are attached to the lateral walls of the segments that are to which the walking legs are attached. Help of this table, and this table presents the branchial formula of the prawn. So what is this branchial formula? Branchial formula is nothing but the gills, otherwise the other gills and other respiratory organs, and their disposition or their position in the prawn's body can be explained with the help of this table. That's why this is called a branchial formula. Now we have if we see on the extreme left, there is a appendages list is given, and here epipodites. Just now we have discussed these epipodites are also accessory respiratory organs. Then actual gills. So these actual gills are podobranchs, the arthrobranchs, and the pleurobranchs. Let us have a look. The 3 pairs of maxillipedes to which epipodites are attached. So these epipodites, 1 pair each are attached to 1 pair of maxillipedes. In the same way, so there are 3 maxillipedes are present, first, second, and third. These epipodites, each pair is attached to these 3 maxillipedes. And then second one. Then second type is the podobranch. So podobranch, we have seen only one podobranch is present, which is attached to the second maxillipede on one side. So the one pair of podobranch attached to one pair of second maxillipede. So totally second maxillipede will be having two respiratory organs, whereas first maxillipede is having only one respiratory organ. Now coming to the third maxillipede, this third maxillipede has the two arthrobranchs attached to it. Two pairs of arthrobranchs attached to two pairs of maxillipedes. So totally, and in addition to that, one epipodite is also present in the maxillipede, third maxillipede, totally making it to totally three respiratory organs are attached to the third maxillipede. So within the maxillipedes, total respiratory organs here in this, totally 6 respiratory organs are present here. Then coming to the walking legs. So I told you there are 5 pairs of walking legs are present here. Walking legs are present attached to the thoracic legs. So here the inner wall of the segments to which these walking legs are attached, on this wall the 5 pairs of gills are attached to each segment, and here one pair of pleurobranch attached to the fourth pair of the first walking leg, the segment to which the first walking legs are attached. The same way, the segments which are having walking legs attached to walking legs, all these segments one pair each pleurobranchs are present. So totally making this 5. So 5 plus 6, totally 11 respiratory organs, among which 8 pairs are gills, actual gills, and 3 pairs are epipodites. In addition to these, we seen that Branchiostegite also functions as a respiratory organ. Now let us discuss the structure of a gill. Just now we have seen the gills, various types of gills within the prawn. Now gills are crescentic in shape. Now we can see here in the figure, we can see the gills are crescentic in shape, that is half moon shape, like this half moon shape, and each gill is attached to the middle. Each gill is attached in the middle to thorax by gill root. There is a gill root is present, and with the help of this gill root is present in the middle, it is attached to the thoracic wall. And through this gill root only, the nerves and blood vessels or blood channels enter and leave the gill through the gill root only. Then what the gill types, how the gill look like in the prawn and other organisms, so other crustaceans, you can see here. So based on the shape of the gills of the prawn are called as phylobranchs. That means they look like leaf. They look like a leaf. Trichobranch in Astacus. Astacus is also called crayfish, where in this the gill filaments project around a central axis. Here you can see, observe in the figure. See here. So the first one is our phylobranch, which are present in the prawn, and the second one, these are called as trichobranch type, which are present in the Astacus, that is crayfish, where the gill filaments project around a central axis like the bristles of a bottle brush. And again third type which is called as dendrobranch type, and this type is present in Peneus monodon, where it is tiger prawn. Here the gill plates are broken into small filaments here in this. So let us have a look at the gills which are present in the prawn, which are called as phylobranchs now. So the phylobranchs consist of 2 rows of leaf-like gill plates arranged like the leaves of a book just now we discussed, and these gill plates are larger in middle and becomes gradually smaller when they go to the ends. Now a longitudinal groove also runs between the rows of the gill plate. Now if we see the transverse section of the gill, so we can see the histological examination. If we take histology of a gill, so the gill plate is made of a single layer of cells. Gill plate, where is this gill plate? Let us have a look at this gill plate. So here you can see this gill plate. So this is the gill plate, and where it possesses a gill base and two flap-like structures, gill plate and flap-like structures, which are called gill plates. This is the gill base, these are the gill plates. So here, if we examine the histology or cells of the gill, so here you can see transverse section of the gill. Here the cells are of two types. They are gill plate is made of single layer of cells within with a thin cuticle on both sides, and cells which are present within the gill plate are called are two types. They are pigmented and transparent cells. You can see pigmented and transparent cells here. So these pigmented cells, transparent are clear cells, which are arranged alternately, and this gill base is present, and these gill plates, this is one gill plate, then this is one gill plate, and here you can see connective tissue is present inside. So the compact connective tissue is present, and then the connective tissue is surrounded by the cuticle, and below that cuticle epidermal layer is present. So connective tissue surrounded by an epidermal layer, and epidermis is surrounded by a cuticle, and these two are lateral longitudinal channels, blood channels. We will discuss later on. And this is the gill base, and this is a median longitudinal channel. Now coming to the prawn respiratory system, blood supply to the gill. How the blood supply? Just now we have seen the structure of the gills, and the types of the gills and structure of the gills. Now coming to the blood supply to the gill. So before entering into before discussing the mechanism of respiration, let us have a look at how the blood is supplied to this gill. First of all, how the blood channels are present in the gills. There are three longitudinal blood channels run through the gill base, two lateral longitudinal, among which two lateral longitudinal channels along the margins, and one median longitudinal channel along the apex of gill base. So here we can see in this figure two lateral longitudinal blood. So just now we have seen the lateral longitudinal channels, how they are arranged, and median longitudinal channel, how these lateral longitudinal channels are connected by transverse connectives, and the transverse connective lying immediately against the root of the gill, gill root is larger and receives blood for aeration from the afferent branchial channel. See here. So here transverse connective, this transverse connective, which is possessing a small opening here, through which afferent branchial channel is connected to this transverse connective, and the lateral channels are also connected with the median channel through marginal channel. Just now we have discussed, and these marginal channels that arise from the lateral channels at the base of the gill plates, and they run along their margin and open into the median channel. Then the blood that enters the transverse connective through this afferent branchial channel enters the transverse connective through afferent branchial channel, branchial blood vessel, passes to the median channel through the lateral and marginal channels of the gill plates. How? Just now we have seen here the afferent branchial channel. So it is connected to the transverse blood vessels, transverse connectives, and these transverse connectives are attached to the lateral channels, and these lateral channels again they are attached to the marginal channels, and these marginal channels again they are attached to the median channel. So all these are connected. So we will look into this how these are interconnected with each other. Then, so just now we have seen all these lateral longitudinal channels and marginal channels and median longitudinal channels and transverse connectives are interconnected with each other. The gaseous interchange between the blood and the water takes place by diffusion during the circulation of the blood through these channels, and the aerated blood or the oxygenated blood is returned by the efferent branchial channel to the pericardial sinus. You can see here the connection with the efferent branchial channel here from the median channel. So whereas the afferent channel has connections to the connectives, transverse connective, and again to the lateral channels and marginal channels, again median channel, whereas the median channel it has connectives with the efferent branchial channel like this. So the blood passes through this course. So now coming to the mechanism of respiration. Before that, let us have a look at the blood circulation in a gill. So in a gill, just now we have seen the clear picture of the interconnectives of the blood vessels and blood channels. Now first of all, let us have a look at the impure blood from the body parts. So blood circulation in a gill, please concentrate here. So impure blood from body parts enters into the afferent branchial channel. All body parts, that means venous blood, enters into the gill via afferent branchial channel, enters into the gill route. Then from where it enters into the transverse connectives. So from the afferent branchial channel, I told you the transverse connectives, and through the transverse connectives the blood enters into the lateral longitudinal channel, and from the lateral longitudinal channels it enters into the marginal channels, and from the marginal channels it enters into the median longitudinal channels, and where through this course the oxygenation is happened. The oxygenation occurs. That means exchange of gases takes place when the blood course or blood transports in this median longitudinal, especially the median longitudinal channel, that means marginal channels. In these marginal channels here oxygenation takes place. So especially these marginal channels the oxygenation takes place, and then the blood from the median longitudinal channel enters into the efferent branchial channel, from where it enters into the pericardium, and where the heart is located. In the pericardium heart is located, and the purified blood or oxygenated blood through the efferent branchial channel enters into the pericardium, from where the ostia open into the heart. Through the ostia, into the heart oxygenated blood enters, and from the heart oxygenated blood is supplied to all body parts. So this is the blood circulation through the gill. Now coming to the mechanism of respiration. So prawn respiratory system, here we have seen so far, in addition to the 8 pairs of gills, there are scapognathids, the branchiostegite, as well as the epipodites are functions as respiratory organs for the gaseous exchange. Now what happens in the mechanism of respiration, how the water is or oxygen is readily available to the gills. So as we know that gills are mainly the aquatic respiratory organs. The gills are mainly the respiratory organs in the aquatic organisms, where the plenty of water should be present to the gills. So how the plenty of water or water current is coming into the vicinity of the gills, let us have a look. Now first of all, the scapognatite makes constant vibrating movements. So what is actually this scapognatite? Scapognatite is nothing but it is a fan-like structure lipids, and this scapognatite that makes the water current possible in the case of the prawn. This capognatite makes constant vibrating movements which create a suction force, and due to this suction force, or actually to maintain a proper respiration, a regular supply of oxygenated water is to be maintained. How this regular supply of water is maintained in the, that is come and available to the gills. A continuous current is set in the continuous current is set and maintained by the constant vibrating movement of this scapognatite of the maxillae. The movements of the scapognatite create a suction force, just now we have discussed, that draws in water, and this water which enters the chamber along the posterior and ventral portions, along the posterior portion and along the ventral margins of the branchiostegite. Posterior and ventral margins are opened because this branchiostegite is covers on the dorsal surface, and whereas the posterior, anterior and ventrally it is opened. So that's why the gill chamber is opened anteriorly, posteriorly and ventrally. So on the dorsal side it is covered by carapace and the branchiostegite. So from the water current, due to the movement or due to the vibration movement of the scapognatite, a water current is created, and this water, fresh water, enters from the behind and ventral sides into the gill chamber. It passes over the gills and then is expelled from the front end and beneath the head region. The epipodites of the maxillipedes supplement this vibratory movement of the scapognatite also, and constant water current is created into the gill chamber, and then the gill plates act as excellent permeable membranes for passage of gases to and fro by diffusion. Thus the gill chamber is constantly supplied with the current of fresh water, and the inner lining of branchiostegites, gills and epipodites are richly supplied with the blood capillaries, and where the exchange of gases takes place. The thin and delicate gills act as excellent permeable membranes for the diffusion of gases, and even inner lining of branchiostegite is also supplied with the blood vessels, and epipodites are also supplied with the blood vessels. They also contribute in the gases exchange, and the dissolved oxygen that is present in the water is taken by the blood, and in exchange the carbon dioxide that is present in the blood is diffused outside into the water. So this is the respiratory system and the mechanism of respiration in the case of prawn. So the prawn is an excellent aquatic organism because of the presence of the gills. Thank you.